Good morning, everyone. My name is Alessandro Sette. I'm a professor of immunology at the La Jolla Institute and co-director of the Center for Vaccine Discovery. Is my great honor to chair this symposium today for the Embassy of Italy Prize on Degenerative Diseases and Aging. Before we start, a couple of words about ISNAF. ISNAF is the Italian Scientists and Scholars in North American Foundation. And the mission, it's a nonprofit organization. The mission is to connect and celebrate and empower the diaspora of Italians intellectually and technologically active in North America, with more than 3,000 members and research and technologists in the network, and promote cultural exchange and integration and with public and private organizations throughout North America. And so today we are here for one of the key aspects of the ISNAF mission, which is to promote and award prizes to young investigators. And the Italian Embassy promotes this activity. The topic of the embassy rotates each year. And so this year, we're focusing on neurodegenerative diseases, including aging. And this is really to underline and stress the ever-growing importance of this topic. So after saying a few words about this, I would like to introduce and thank the members of the jury, Professor Maria Cristina Alberini, which is professor of neuroscience at NYU, Professor David Hafler, who is the chair of neurologist in chief at Yale, New Haven Hospital, and Ekemini Riley, who is the president of Coalition for Us Aligning Sciences and the director of Aligning Science Across Parkinson, or ASAP. So many of you may know of ASAP, really playing an important role. If you have questions for the attendees, put questions in the chat, and there will be questions at the end of each talk from both the jury and the chat, depending on time, of course. We will have 3 presentations, 15 minutes each, and then the jury will decide the award on a separate session. So the finalists are Simone Brioschi. He is an instructor at WashU in St. Louis in the School of Medicine. And then there is Francesca Garretti, is a postdoc in the Department of Genetics and Genomics at Icahn School at Mount Sinai. And Roberta Peruzzo, a postdoc at UC Berkeley, pardon me, molecular and cell biology. And so we will go in alphabetical order. So we will start with Dr. Brioschi, who will tell us a study of brain immune cells during development, homeostatic in disease. The floor is yours. Thank you.
Okay, do you hear me?
Yes, now we can hear you.
Okay, perfect. Okay, thank you very much, Dr. Sette, for this very great introduction. And I also want to thank the ISNAF for organizing this meeting and giving me the opportunity to share some of my data with you today. So as you can see from the front slide, I'm really interested in the brain macrophages. These are very important cells under steady state and also during disease. And they can be very important also during neurodegenerative diseases, which is basically the core topic of this session. Before we dive into the data, let me give you some insights about the cells. You probably heard about the microglia, which are the resident macrophages in the CNS parenchyma. And these cells represent the majority of the myeloid cells in the brain. However, if you look at the brain border region, you can find different subsets of brain macrophages, which are very different from microglia. And in particular, I'm talking about the perivascular macrophages, the choreoplexus macrophages, and the meningeal macrophages. These subset of macrophages at the brain borders are collectively referred to as brain border associated macrophages, or BAMs. So let's have a look how these cells originate and how they reach the brain. What we know now is that the CNS macrophages originate during the embryonic development, and more specifically from the yolk sac hematopoiesis. And the earliest progenitor of these cells are this progenitor called erythromyeloid progenitor, which generate myeloid cells that trafficking through the embryonic circulation reach the brain, and they locally differentiate into either microglia or BAMs, depending on their location. So let me give you some bullet points. We now know that microglia and BAMs derive from a unique progenitor, and the specification of yolk cell-derived macrophages into microglia or BAMs occurs locally into the CNS niches. However, signal and transcription factors that are orchestrating the development of the cells are incompletely understood, and this is what I want to study. So why do we care where the cells so important? Well, brain macrophages are very important in homeostasis, for example. Microglia have been shown to be remodel or shape the synaptic connectivity between the neurons, or the perivascular macrophages are important in regulating the dynamics of the blood vessel, for example, regulating the deposition of the collagen. However, these cells are also very important during disease. Microglia phagocytose the amyloid beta during amyloid pathology. And these cells can also produce inflammatory cytokines and are involved in the tissue repair during neuroinflammatory condition. So if we want to understand this broad spectrum of different functions, we need to clarify how the cells develop and how they become specialized within their environmental dishes. And this brings me to an outstanding question for the field. So what are the mechanisms that are dictating the fate of the microglia and the BAMs? There might be different transcription factors or different epigenetic modification, or probably both. However, if we want to investigate a particular molecular mechanism during the development of the cells, we need to target embryonic macrophages highly efficiently and specifically, and especially very early during their development. And here there's a technical limitation, because at the moment there are no Cray lines that allow for a specific and efficient targeting of CNS macrophages in development. There are Cray lines that can that are used in this microglia research. However, none of this has these three important features: efficiency, specificity, and early activation. So to start this project, I wanted to generate a new tool to investigate the development of CNS macrophages in more detail. And to do so, I have to find a microglia-specific gene, or a gene highly expressed in brain macrophages, specifically and early during development. So I started to search in different gene expression data sets that are publicly available. For example, these are all different immune cells analyzed in the Tableau Muris project. And as you can see, I found this gene, CRIB1, which is highly expressed in microglia. More importantly, CRIB1 starts being expressed in the brain as early as E13.5, and the expression level remains highly stable during the perinatal period, and then is downregulated later after birth. So potentially, CRIB1 is a good candidate to make a new tool to target these cells during development. And this is what we did. We introduced the Cray sequence downstream the CRIB1 promoter, generating the CRIB1 Cray, which was then crossed with the ROSA26 tomato to assess efficiency and specificity of recombination. And here you can see a representative FACS plot of brain macrophages. We have the microglia and the BAMs. The recombination in microglia was basically 100%. And we also had about 30% recombination in the BAMs. And unfortunately, in the interest of time, I cannot go too much into the details. However, in the paper, we show that these populations here are the BAMs that specifically originate from the embryonic hematopoiesis and not from monocytes. So then we generated a new tool, which we carefully characterized, and then we wanted to use it to answer a biological question. And we decided to focus on the TGF-beta signaling. So the question is, what does TGF-beta signaling do on the development of the cells? We chose TGF-beta because it has already been shown before that TGF-beta is important for the microglia development. However, TGF-beta receptor activate two distinct and non-overlapping pathways. For example, there's a canonical pathway, which requires phosphorylation and dimerization of SMAD protein, which then bind at SMAD4 to migrate into the nucleus and activate the TGF-beta transcriptional program. In parallel, TGF-beta receptor activate a non-canonical pathway, which mostly relies on MAP kinases. So at the moment, the specific contribution of the canonical and non-canonical pathway to microglia development is not known. And to investigate this, we decided to knock out SMAD4 specifically in brain macrophages during development using our new tool. Then we performed single-cell RNA sequencing of brain immune cells. Here you can see in this cloud, these are microglia from the SMAD4 flox mice, whereas in this cloud here, there is the microglia from the SMAD4 conditional knockout. As you can see, microglia from the two genotypes split on completely separate UMAP territories, indicating broad transcriptomic changes in the cells, whereas the clustering of the BAMs remained unaffected. So next, we inspected the differentially expressed gene in more detail, and it was immediately clear that microglia without SMAD4 exhibited a broad downregulation of all microglia signature genes. Microglia under steady state express a set of genes that altogether shape the so-called microglia signature, such as SPARC, CELPLG, XB, TMEM119, and P2 receptor Y12. However, if you remove SMAD4, the expression of this gene is lost. And this phenotypic transformation can be exemplified in this FAX plot, where you can see that a wild-type microglia expressed TMEM119 and P2 receptor Y12. Whereas if you knock out SMAD4, this population disappears. Even more interesting, microglia without SMAD4 upregulated several genes of the BAM signature. There are several genes that are not expressed in microglia in steady state, but they are always expressed in BAMs, such as ApoE, MARC1, LIGHTS2, PF4, MS487. So this gene, microglia do not express this gene, but if you knock out SMAD4, they suddenly become positive for all these markers. And here you can see a representative example. CD206 is a typical marker of BAMs, which is not expressed in microglia in wild-type mice. However, if you remove SMAD4, microglia become CD206 positive. So altogether, this data indicate that if you delete SMAD4, microglia undergo a phenotypic change, a phenotypic shift from the homeostatic signature towards the BAM signature. Next, we perform also single-cell ATAC to assess the chromatin accessibility landscape. And long story short, this analysis shows that if you remove SMAD4 from microglia, this cell acquire an epigenetic signature which is indistinguishable from the BAMs. For example, we can have a look at this microglia marker gene, which is TMEM119, and these peaks here represent the region of open chromatin. As you can see, the locus is highly accessible in the wild-type microglia. However, if you remove SMAD4, the chromatin becomes less accessible, just like in the BAMs. Conversely, if you look at the BAM signature gene like ApoE, the locus is not accessible in wild-type microglia. However, if you remove SMAD4, the chromatin opens up, similar to the BAMs. By contrast, the BAM chromatin landscape was unaffected between the two genotypes. So altogether, this data indicates that if you remove SMAD4, microglia fail to differentiate and maintain and acquire a transcriptomic and epigenetic signature, which is very similar or indistinguishable from the BAMs. So next, we perform a large battery of behavioral tests. We perform tests for locomotor activity, exploratory behavior, and coordination. And we did not find any difference. This is, for example, this is the accelerating rotor rod assessing motor learning. And as you can see, there is no difference between the genotypes. However, we did find significant differences at the Morris-Potter maze, which is a task assessing learning and memory. And this is the latency time to reach the hidden platform. And as you can see, the SMAD4 knockout mice do not learn the task. And they also spend significantly less time in the target quadrant, indicating severe memory impairment. So I can summarize what I showed you so far. We generated a new tool to target CNS macrophages during development with very high efficiency and specificity. Using this tool, we showed that SMAD4 is required for microglia, but not for the BAM specification program. And additionally, SMAD4 deficient microglia become transcriptionally and epigenetically akin to BAMs. And more importantly, mice with SMAD4 deficient microglia exhibit memory impairment, emphasizing how important these cells are. If you block the development of microglia early during development, you can affect brain functions. This is all published, and now I would like to share with you some data from the follow-up project that I'm working on now. So we showed with this all this work, we showed that SMAD4 is important for microglia, but not for the BAMs. And then the next question is there any transcription factor that is controlling BAMs development, but not microglia development? So to check this, I went back to my single cell data. These are the single cell RNA-seq from brain macrophages. The big population are microglia. And then we have two distinct subsets of BAMs, which I labeled as BAM2 and BAM1. BAM2 are the BAMs that are specifically of embryonic origin, whereas the BAM1 are monocyte-derived. Then I started to search for transcription factors that are differentially expressed between this population. And I found an interesting candidate, which is MAF, which appeared significantly enriched in the embryonic-derived BAMs, but not in the compared to the other two populations. So then I checked also our single cell ATAC data. And here, this plot shows the motif accessibility for different transcription factors. So positive value indicate that the transcription factor has DNA binding motif enriched in open chromatin region. And as you can see, MAF and C binding protein appear as the most enriched transcription factor in the BAMs compared to microglia. So let's try to integrate the transcriptomic and the epigenetic information, and we generated this plot. On the x-axis, we have the log2 fold change of the gene expression. So positive value indicate that the gene is highly enriched in the BAM compared to microglia, whereas on the y-axis, there's the binding motif accessibility, and the positive value indicates that the motif is highly accessible in the BAM compared to microglia. Doing so,
2 more minutes to go.
Yeah, I'm almost done. Doing so, we restrict our the number of hits to only one good candidate, which is MAF. So then we generated MAF conditional knockout using our newly generated tree, which knocks out MAF from both microglia and BAMs. And here is very interesting, because if we gate on the BAMs, you can see this population here, which is positive for CD1 to 63 and negative for class 2. These are the BAMs that are specifically embryonic derived, and this population disappear if you remove MAF. We also took some images. Here you can see in white the brain blood vessel, and in red is the staining for CD206, which is a typical marker of embryonic derived BAMs. And you can see the cells are very abundant on the brain surface and also on the perivascular space. However, if you remove MAF, these population disappear and they are replaced by class 2 positive macrophages. I can also show you a closer look at these cells. These are perivascular macrophages in the MAF flux mice, which are CD206 positive but do not express class 2. However, if you remove MAF, the CD206 is downregulated, and you see this dramatic expansion of class 2 positive macrophages. So this is the take-home message. Yolk-sac-derived macrophages migrate into the brain during embryonic development. If they activate SMAD4, they become microglia, whereas if they activate the MAF, they become BAMs. So if you delete SMAD4, there's a failure, microglia development, which causes memory impairment, whereas if you delete MAF, there's an arrest in the BAMs development, and the consequences for the brain are currently under investigation. So with this, I would like to thank all the people that participated in this project, especially my colleague from the Colonial Lab and our funding sources, and also thank you to the ISNAP for organizing this beautiful event. And with this, I want to thank you for your attention, and we can open the discussion. Thank you.
Okay, thank you, Simone. I would like now to invite members of the jury if they have questions to ask directly.
I do, if I may. So one of the things we're finding as we do spinal taps in patients with neurodegenerative diseases are what we're calling CSF macrophages, which have ApoE4, C1q, TREM2, other markers of complement. So to tie your work to the human disease, how would these BAMs or other cells, well, I mean, the CSF cells we're seeing that have these non-classical myeloid characteristics, how would they be represented in your mouse work? Where do you think they come from? I don't think the yolk cell.
This is a this is a very interesting question. At the moment, no one really knows the composition of the or the origin of the BAMs from the inpatient, especially during disease, and this is something that definitely needs to be investigated. These cells are relatively few, and especially collecting tissue from postmortem patient, the majority will be microglia, and the BAMs are relatively few cells. So of course, it's easier to get many cells from mice, but from patient it's definitely less feasible. I wouldn't be surprised if monocytes gave an important contribution to this population during disease. Talking more specifically about the translational approach of this of my model, we are now trying to knock out genes that in human contain polymorphism that are linked to neurodegenerative diseases, especially Alzheimer, and see how this how the Alzheimer's disease or amyloid pathology evolves if we knock out this gene specifically in brain macrophages.
Thank you.
Yeah.
I have a quick question. I'd like to jump in here. Simone, you gave a very great presentation. My question is about how reversible these outcomes are. Have you all explored any reversal experiments to see if, reconstituting SMAD4 leads to a reversal of the transcriptional program that you described? Do they revert to be more microglia-like, or are they already faded?
This is it's a very interesting question. So if we can induce an overexpression of, for example, SMAD4 or other components of the TGF-beta signaling in the BAMs, if they become more microglia-like, this is basically the question.
Yes.
I have not tried that yet. I tried to induce MAF overexpression in microglia, but in culture so far.
Okay.
And yes, we see upregulation of genes, of BAM signature genes. However, the model is, it's suboptimal because microglia in culture are very different from the microglia in vivo in the brain. So the conclusions of this type of experiment are, let's say the data need to be carefully interpreted and in the proper context. This is an experiment we could easily do. We could induce MAF overexpression, for example, put MAF under the control of a strong microglia promoter or into a ROSA26 locus, which then gets activated upon recombination, and see whether the microglia become more BAM-like. Concerning the TGF-beta, okay, SMAD4 is important, but the cell needs to receive the upstream signal, which is TGF-beta. So I'm not so sure how much TGF-beta is present in the extra parenchymal compartment. Yeah.
Nice. Thank you.
Yeah. Thank you for the question.
Simone, a quick question from me. I missed how you check the specificity of this SMAD4 genetic approaches, because obviously SMAD4 as well as TGF-beta is very important for neuronal differentiation, learning and memory plasticity, and so on and so forth. So maybe I missed it. How did you exclude that there is an effect on neurons?
Because we test the recombination specificity using the ROSA26 tomato reporter, and then we check also in the brain and also in different myeloid cells from the periphery, and the recombination was highly efficient and specific for microglia. There were very sparse recombination in some OPCs and some neurons mostly in the cerebellum, but very few cells. And this is mostly, this is presumably a leakage of the ROSA26 tomato. We can probably investigate, do more control to make sure that SMAD4 has not been deleted in other cells. But for sure, the recombination in microglia was 100%.
Okay, thank you. Go ahead. I was going to ask if there are any questions from attendees or any additional questions.
Apparently not.
Okay, well, hearing none, then I think we can move to the next presentation, which I trust is going to be from Francesca Garretti from Icahn School of Medicine at Mount Sinai. And I think the title was Investigating the Impact of Disease Risk Modifying Variants of Microglia Function. Francesca.
Thank you so much. Thank you, Dr. Sette, for that introduction. And first of all, I'd like to thank the committee for inviting me as to give a talk and for nominating me as a finalist for the 2023 Young Investigator Award, Embassy of Italy. I am Francesca Garretti, and I am currently a postdoctoral fellow at the Icahn School of Medicine at Mount Sinai. And I'll be giving you guys a talk about recent work that I've been working on as a postdoc in the lab in the past 2 years. It's all unpublished and work in progress. So wanted to show you what we've been working on. And we've been looking at investigating the impacts of Alzheimer's disease risk modifying variants on microglial function. So Alzheimer's disease is a progressive neurodegenerative disease, and it's characterized clinically by memory impairments and cognitive decline. As the disease progresses, you can see in these coronal sections, there is substantial brain atrophy in advanced AD brains in comparison to healthy controls. The disease was first described by Alois Alzheimer's in 1907, and he also had reported some hallmarks of the disease that still hold true today. These are neurofibrillary tangles of hyperphosphorylated aggregated tau, as well as plaques of amyloid beta. In addition, you also have microgliosis, so microglial proliferation, as well as lipid accumulation and lipidosis. Much progress has been made since then in identifying the variants that contribute to Alzheimer's disease risk. The first to be identified were these familial autosomal dominant mutations in APP and presenilin. These are very rare in the population, however, they're fully penetrant. And they were very helpful in trying to understand the pathophysiology of amyloid deposition and aggregation, as well as the enzymes involved in it. However, now the attention has shifted to genome-wide association studies to try to understand and identify variants that contribute to Alzheimer's disease risk. These variants are of varying frequency in the population in terms of rarity and commonness, and they have differing effects on AD risk. You can tell that there are many common variants that have a slight effect on AD risk. However, here the ones that stick out the most are ApoE4 variants. ApoE variants. So ApoE4 increases AD risk. This is on an absolute scale and in a dose-dependent manner, while ApoE2, on the other hand, decreases AD risk as well in a dose-dependent manner. So ApoE, what is it? It is an apolipoprotein, and it is involved in microglial efferocytosis of cholesterol-rich cellular waste. So it is a way that cells mitigate cholesterol-rich accumulation from myelin fragments and apoptotic cells and digest it and secrete excess lipids. ApoE is involved in the recognition of cholesterol-rich waste, as well as the secretion of excess cholesterol from lipid droplets. ApoE comes in 3 different isoforms that are determined by 2 amino acids in 2 variants. And these variants have an effect on the conformational structure of ApoE. So ApoE2, which decreases AD risk, comes in a more open conformation that renders it a more accessible lipid binder, while ApoE4 is more closed in, and so it is less of an effective lipid binder. As I mentioned, ApoE dosage increases AD frequency in the population, as well as decreasing the mean age of onset of Alzheimer's disease. However, we know that not all ApoE4 carriers eventually end up developing Alzheimer's disease, and there's a quite wide range of the effects of ApoE4 on patients. This suggests that there are additional factors that could be mitigating the ApoE4 risk on Alzheimer's disease, and that's exactly what we sought out to answer. So within the lab, we ran a stratified GWAS study with 2 very distinct populations of ApoE4 carriers. The first population is young AD cases that are ApoE4 carriers. So these are young Alzheimer's disease patients that are 65 years and younger and carry at least 1 allele ApoE4, in contrast to cognitively normal ApoE4 carriers. These are patients that are 80 years or younger and are cognitively normal, even while carrying ApoE4 alleles. This is to identify any variants that are increasing risk for Alzheimer's disease in the young AD cases, or protecting patients from Alzheimer's disease in the protective variants. What we found was an association with eIF2B3 in the young AD cases. This signal was largely led by an association with this SNP that is actually amino acid. It causes an amino acid substitution at 4,4 from a serine to an alanine. This was an interesting finding in the lab because we had previously identified a novel association with a similar gene, eIF2B2. In this published study, myeloid cell genomics and AD genetics were integrated to identify AD risk variants, and we found a novel association with eIF2B2 with Alzheimer's disease. At the time of publication, the directionality was unknown, but we've conducted since then preliminary studies to identify that eIF2B2 is associated with increased AD risk by lowering eIF2B2 signal. So what are eIF2B2 and eIF2B3? They are subunits of eIF2B, which is a key enzyme involved in translational control and the integrated stress response. So to begin to understand this pathway, we turn to eIF2. eIF2, in its active GTP-bound form, binds to initiator tRNA and allows for a ribosome assembly and protein translation initiation. This expends energy from GTP to GDP, and then eIF2B continues this pathway by exchanging the GDP to the GTP. Under various types of cell stress, such as viral infection, ER stress, or various types of starvation, you have the activation of kinases that phosphorylate eIF2. Phospho-eIF2 is now no longer a substrate, but rather an allosteric inhibitor of eIF2B. What this does is it shuts down global protein synthesis and allows for the translation of cap-independent transcription factors, such as ATF4. This in all is the integrated stress response, and the goal of it is to restore cellular homeostasis, and if that cannot be reached, to induce cellular apoptosis. So mutations in any of the 5 subunits of eIF2B have been shown to cause vanishing white matter disease. Vanishing white matter disease is a childhood onset demyelinating disease in which the clinical manifestation is exacerbated when a patient is subjugated to increased stress. We have looked at the role of translation in the context of ApoE4 at the transcriptomic level in the lab. So in the first study, we saw that protein translation as well as elongation and initiation is actually down-regulated in ApoE4 for mouse microglia compared to ApoE33 microglia. In comparison, in iPS-derived human microglia, you have a downregulation of protein translation in ApoE4 for microglia when you compare it to ApoE33. In addition, EIF2 signaling in the ISR pathway is implicated in Alzheimer's disease when other AD risk genes are decreased or increased of their expression. So if you alter PU1 or TREM2 expression, you have differential expression of the ISR pathway. In addition, when the ISR pathway is corrected in AD mouse models, you have a correction of memory impairment in these mice. So what we wanted to address first off, and what I'm going to show you today, is how the integrated stress response is impacted in these two genetic backgrounds. So an ApoE4 background, as well as reducing EF2B3 and EF2B2 genes. The first we do in iPS-derived human microglia, and the next in human macrophages. So in iPS-derived microglia, we have 5 different ApoE33 and 5 different ApoE44 donors that we use to differentiate from iPSCs to microglial-like cells. In culture, we treat them with a methionine analog, and in the presence or absence of thapsigargin, which is a integrated stress response inducer. After a short window of time of 3 hours, we collect the cells and perform click chemistry to conjugate a fluorescent dye to the methionine analog to give us a readout of protein translation, in addition to looking at markers of the integrated stress response. What we found in ApoE44 microglia at baseline is that there is a significant reduction in protein translation in ApoE44 microglia when compared to ApoE33 microglia. When we look at cells in the context of the integrated stress response with thapsigargin treatment, we see that thapsigargin was successful in reducing protein translation levels in ApoE33 microglia, and this pattern is absent in ApoE44 microglia, likely due to, we postulate, a basement effect of protein translation inhibition. However, when we look at different markers for the integrated stress response, so looking at phospho-EF2 alpha over total EF2 alpha, as well as ATF4, we see that the integrated stress response is maintained in both genotypes of microglia, and there's no significant difference between the two. Next, we wanted to look at the effects on the integrated stress response by modulating EF2B2 and EF2B3 expression in human macrophages. Similarly, we deplete the media of methionine and introduce a methionine analog in the presence and absence of thapsigargin. And similarly, collect the cells to perform click chemistry to look at protein translation. What we find is that at baseline, there's a significant reduction in protein translation in EF2B2 knockdown macrophages compared to the scrambled controls.
2 more minutes.
Yep. When the cells are treated with thapsigargin, we see that there is a significant reduction in protein translation in all genotypes. However, there's a maintenance of the difference between EF2B2 and scrambled cells. Next, we looked at markers of the integrated stress response, and we see that at baseline, there's a baseline activation of the ISR in EF2B2 knockdown cells, and thapsigargin increases these signals in all genotypes. However, we see that there is an interaction effect in EF2B2 knockdown cells when compared to scrambled. This means that thapsigargin has a significantly different effect on EF2B2 knockdown cells compared to the controls. So what I've shown you so far is that the background and previous literature has implicated EF2B and the integrated stress response with Alzheimer's disease. And what we found is a novel association with EF2B2 and EF2B3 as novel risk variants for Alzheimer's disease. Looking at the effects of the integrated stress response, we see that ApoE44 microglia have reduced protein translation at baseline, but an intact integrated stress response, and reducing levels of EF2B2 and EF2B3 dysregulate the integrated stress response in human macrophages. Our next step, and what we're currently working on right now, is to take this 5 ApoE44 IPS lines that we have and reducing EF2B2 and EF2B3 levels with CRISPR-Cas9 technology to look at the effects in vitro. In addition, what we do in the lab is that we have microglial progenitor cells that we transplant into humanized immunodeficient mice that are in a wild-type background as well as in a 5X FAD background. And these cells, these microglial progenitor cells, then repopulate the mouse brain. This allows us to look at human microglia in the context of in vivo context, but also in the background of Alzheimer's disease. And with that, I'd like to thank my lab and everybody that's involved in the project, our collaborators at WashU and at UCI, our funding sources, and all of you for listening. Thank you so much.
Thank you, Francesca. I would like to open the forum to the members of the jury for questions. I see a raised hand from David Hafler.
Yes, but both talks are beautiful, and we can't wait for the third one. So one of the things that's emerging in terms of Alzheimer's and microglia function is TIM3, which is one of the genetic hits in Alzheimer's. Now, I don't expect you to know every gene associated with this, but it may be more suggestion than a question in all fairness. But there's a paper that's going to be coming out from our dear friend and colleague of Alex and mine, Vijay Kudru, that TIM3 knockouts the microglia can process the amyloid. So if you haven't looked at TIM3 in terms of the just genetic variant, it might be worth looking at.
Oh, that's a great suggestion. Yeah, so the way there are many genes, especially, but what's interesting is how there's a convergence to microglial function also in terms of dysfunction in effects on Alzheimer's disease pathology.
The TIM3 may end up being a particularly important, at least from an immunologist point of view. So exactly. Anyway, beautiful work.
Thank you.
No questions from me? Thank you.
Yeah. Anybody else?
Alex, you're muted.
Sorry, I say, hearing no additional questions from the jury or any of the other attendees, I think we can move to the third and last talk from Roberta Peruzzo, a postdoc at UC Berkeley, and the title that I had, it was Mitochondrial and Lysosomes as Therapeutic Targets in Neurodegeneration. At least that was a provisional, yeah. Roberta, the floor is yours.
Thank you. Can you see my screen?
Yes.
Okay, so thank you so much for the kind introduction. I would like to thank the scientific committee for selecting me as a finalist for this prestigious award. As you can see in the title, today I will speak about mitochondria and lysosomes that are two key organelles for maintaining the neuronal homeostasis. Neurons are the fundamental units of the nervous system, highly specialized cells that play a pivotal role in transmitting and processing information through electrical and chemical signals. Neurons depend on the correct function of both mitochondria and lysosome for maintaining their homeostasis. Being post-mitotic, neurons are extremely reliant on the optimal function of their autophagy and lysosome system for degrading damaged organelles and protein aggregates and ensuring the neuronal survival. On the other hand, also mitochondria, through the oxidative phosphorylation, produce the energy in the form of the ATP that is essential for sustaining neuronal processes and in particular the synaptic activity. So mitochondria and lysosomes also interact to guarantee the correct neuronal homeostasis. And most importantly, their dysfunction contribute to the development of neurodegenerative diseases. Neurodegenerative diseases are a group of neurological disorders characterized by the progressive deterioration of nerve cells, leading to cognitive and motor dysfunction. Among these, I want to mention the most present that are Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis. And among all the hallmarks of neurodegenerative disease, mitochondria don't produce enough energy to sustain the neuronal system, and lysosome, on the other side, do not clear protein aggregates that are a feature of the presence of the neurodegenerative diseases. Among all the neurodegenerative disorders, my research focused on amyotrophic lateral sclerosis and frontotemporal dementia, that are two devastating progressive diseases which involve the degeneration of neurons that are essential for muscle movement in the case of LS, and are essential for cognitive behavior in the case of frontotemporal dementia. And the lysosomal and mitochondrial homeostasis is impaired in these disorders. And focusing on the pathogenic point of view, different genetic mutation in quality control genes result in a shared pathogenic hallmark, that is the aggregation of TDP43 protein in the brain and in the spinal cord of the patients. And these aggregates escape the autophagy clearance. So as a result, on one side, boosting autophagy and lysosomal function, or on the other side, restoring mitochondrial activity, may be two important promised routes to degrade TDP43 aggregates and restore the overall neuronal homeostasis. So building on that, the aims of my project are to understand and restore lysosomal and mitochondrial function in FTD-LS. So as first, in collaboration with Regina, that is another postdoc in the lab, I generated this powerful system to differentiate neurons starting from iPSC and iPSC from FTD patients. In this way, what I can do is to study the mechanism that underline lysosomal and mitochondrial dysfunction and the potential therapeutic strategies in this relevant disease model. But before going to detail, I need to briefly explain the mechanistic basis of lysosomal dysfunction. So lysosome is the center of activation of mTORC1, that is the master regulator of cell growth and metabolism. After the phosphorylation of a plethora of different substrates, mTORC1 activate anabolic processes and inhibit catabolism. And to do that, it acts directly, inhibiting the initiation of the autophagosome formation. But on the other hand, also it acts at the transcriptional level, inhibiting TFAB. TFAB is a transcription factor that is the master regulator of lysosomal biogenesis and autophagy. When it is active, it sustains the positive feedback loop in the catabolic processes. However, once mTORC1 inhibits TFAB, TFAB resides in the cytoplasm and it's no longer able to activate the transcription of the downstream genes. So in this lysosomal context, I first analyzed how the autophagy lysosome system is regulated in neurons differentiated from patients. What I observed is that mTORC1 signaling is hyperactive in patient cells in comparison to the health donor, as you can see in this Western blot. And of course, if mTORC1 is hyperactive, what we expect is that TFAB is in the cytoplasm and is phosphorylated. And indeed, as you can see in this graph, by the decrease of the transcription of the TFAB target genes, TFAB was inactive. So building on that, patient cells saw both the upregulation of mTORC1 and the downregulation of catabolism. So basically, you're restoring the correct autophagic function would be a good strategy to tackle FTD. So what are the strategies that we can use to upregulate autophagy? Starting with the more intuitive thing that we can do, the first way to upregulate autophagy is to pharmacologically manipulate mTORC1 itself. To do that, in our lab, we have already developed a new generation of mTORC1 specific inhibitors, which act in a different way than the traditional mTORC1 inhibitors, because the traditional inhibitors, like rapamycin, for example, are not indicated for the treatment of the neurodegenerative disease because they cause side effects, in particular immuno side effects. So what we can do is to test the capability of our new inhibitors, in particular EN6, that is present here, to induce autophagy in vitro. And we've shown that EN6 induce both the activation of TFAB, as shown by this beautiful nuclear localization of the transcription factor, and the clearance also of the TDP43 aggregates, as you can see in this panel. However, all these experiments were performed in vitro in a non-neuronal model. So for this reason, what I'm currently doing is to test these novel inhibitors in the neuronal system that I set up. So taken together, I've shown you that the global inhibition of mTORC1 led to the degradation of protein aggregates, but however, as you can see in this figure, mTORC1 play many roles in the cells. And some of them are very important for neurogenesis, for example, neuronal development and also synaptic plasticity. So for this reason, discover new drugs that specifically act on the catabolic arm of regulation of mTORC1 will be a promising strategy to selectively boost autophagy without interfere with the regulation of the anabolism. Okay, so in this context, my own work contributed to discover the role of two key players of the mTORC1 pathway that are called follicular and FNIP. Follicular and FNIP regulates the nuclear localization and the activation of TFAB. And what is important to notice is that this complex is completely dispensable for the mTORC-dependent regulation of anabolism, but is absolutely necessary to allow mTORC1 to phosphorylate TFAB. So as a result, in normal condition, TFAB resides in the cytoplasm, and the transcription factor of these genes is inhibited, as you can see in this picture. However, when folliculin is deleted, TFAB is no longer recruited on the lysosomal membrane and not phosphorylated by mTORC1. So as a consequence, TFAB is active and localized in the nucleus, where it regulates the transcription of several catabolic genes. It's also important to notice that in order to induce TFAB nuclear localization, it's not necessary to eliminate completely the protein, because as you can see in this part of the panel, a point mutation that is able to disrupt the function of folliculin is sufficient to drive TFAB nuclear localization. So for this reason, discovering novel specific inhibitors of the follicular and FNIP activity is a promising strategy to induce TFAB nuclear activation and autophagy upregulation. To pursue this goal, I set up a microplate-based assay to perform a high-throughput small molecule screening in order to efficiently screen multiple compounds in a short time frame. So very briefly, I'm using a very robust colorimetric assay already developed in the lab as a readout. This assay tests the enzymatic activity of folliculin, monitoring the activity of the folliculin over time. So using this method, I will be able to find potential small drug candidates that can inhibit completely the activity of folliculin, as you can see in this example, in this blue one, or just partially, as the red one, blocking the reaction. So the screening is on the way in the lab, and I will test the potential drugs in neuron in order to detect their capability to boost the clearance of the neurotoxic aggregates. So moving forward on the mitochondrial side of the talk, further investigation performed in the neurons differentiated from patients showed also an altered mitochondrial metabolism with a dysregulated expression of some intermediates of the Krebs cycle, and on the other side also the upregulation of the glycolytic pathway. And all of them are hallmarks of energy imbalance. Luckily, the work that I've done during my PhD put me in a good position to find a novel strategy to overcome the mitochondrial dysfunction that characterize patient cells. In particular, during my PhD, I studied genetic diseases due to deficiency in the mitochondrial respiratory chain complexes, and in particular in complex 3. These mitochondrial diseases are all characterized by a dysfunctional activity of the electron transport chain that induce a reduction in the amount of the ATP production, generating also dysfunctional neurodegeneration and dysfunctional activity and dysfunctional movements of the patients. What is really important is that this condition is extremely similar to the mitochondrial dysfunction characteristic of the neurodegenerative diseases. So in this context of the genetic disorder, what I've done is to use pyocyanin, that is a bacterial Redor cycle, to restore the electron transport through the respiratory chain complexes. In particular, what I demonstrated is that pyocyanin is able to restore the mitochondrial membrane potential, as you can see in this black line, and also the respiratory rate in patient cells, increasing also the production of the mitochondrial ATP, as you can see in this slide. So I tested different kind of patient cells, and all of them had the same phenotype. So building on that, we decided to try it in vivo. And as a consequence of all these results, pyocyanin also very strikingly ameliorates also the locomotor performance in different in vivo models of mitochondrial deficiency.
2 more minutes.
Okay. And in this movie, I want to show you the amazing effect in zebrafish. The test that I'm going to show you is called the evoked touch response test, and it determines the acceleration of swimming movements, which is a proportional measure of the muscle force of the fish. So pay attention on the small fish that is going to be present here in the center. So in response to a mechanical stimulus, such as a small tap, wild-type zebrafish exhibit a fast swimming action. But as you can see now, mutant zebrafish basically do not move at all. And now is going to be the exciting part. Mutant zebrafish treated with pyocyanin recover the muscle force. And as you can see here, after touching the fish, they start swimming again. So we were really happy about these results. And of course, we can apply also these particular molecules, not only in mitochondrial deficiency, but also in other diseases that are characterized by the impairment of the mitochondrial activity. So in summary, I have the expertise and the models to both act on the lysosome and mitochondria to restore the correct functionality of these organelles, which is compromised in ALS and FTD. And what I'm doing, I'm following basically different strategies to do that. I'm trying to upregulate autophagy using compounds that globally inhibit mTORC1 activity, or also small molecules able to induce the selective activation of the transcription factor TFAB. In addition, I can also possibly integrate this strategy with the restoration of the mitochondrial function using biocyanine. So in the future, I'm planning to validate the use of these novel inhibitors in both neuron differentiated from patients-derived iPSC cells and also in vivo in mice models of neurodegenerative disease, and alone or in combination with biocyanine in order to restore the proper neuronal homeostasis and maybe hopefully expand the therapeutic potential for these neurodegenerative diseases, especially FTD and ALS. And finally, I want to thank my mentor, Roberto Zoncu, who is an amazing mentor and helps me a lot every day in order to improve myself. I want to thank all my colleagues, especially Regina, who is collaborating in this project. I want to thank also the funding agency that helped my research, especially IARC, which allowed me to move to the United States and to have this amazing opportunity to work in the special environment of Berkeley. And finally, I want to thank all of you for the kind opportunity and for your attention.
Thank you, Roberta, and open the floor for any questions from the jury.
I have a quick question. So Roberta, if I'm understanding correctly, your restoration of mitochondrial function is dependent on complex 3, right? Based on what you've shown?
So the molecule that I shown acts on both. It's not completely dependent on complex 3. So
okay.
Let me show you the slide. So basically this molecule is a redox cycle and it's able to accept electrons from both NADH and ubiquinol and to donate them to cytochrome C. So what this molecule does is basically bypassing complex 1 and complex 3 that usually is compromised in different kind of genetic disease in this case, but also in different kind of neurodegenerative disease, also in Parkinson's disease.
Lovely. Okay, great. I was going to ask how you rule out contribution from the other complexes in the electron transport chain, but I see that that is not ruled out. So thank you.
Roberta, I have a question. Since this is all about, first of all the talks were amazing. Thank you, all three of you. So going back to the question, I see this is directed toward, of course, the development of novel therapies, perhaps a combination of these steps that are impacted in neurodegeneration. I happen to work on learning and memory disorders, so I'm particularly interested, of course, in the development of novel therapies for cognitive impairments. And my question to you is, besides rapamycin, what has been tried to as a potential therapeutics that can support the idea of activating autophagy in FTD? Was something else tried? There are, there are other things available that should be tried, I think. Do you know?
So there is another, there are some therapeutics that are going, that are in trial right now, not in trial, actually, they are using and they are having a very good response on patients, but they are mainly for ALS, for example, and is basically the use of oligonucleotide, oligonucleotide inside the cells in order basically to inhibit the expression, maybe. So it's basically a target treatment, a genetic treatment, in order to inhibit the expression of particular proteins that are involved in the disease. An example is, for example, SOD1, that is mutated in many of these diseases. And using this kind of gene therapy has shown very important progresses in this field. So I think this is one of the, this is very promising. And actually, it's a thing that we can also do using folliculin, for example, because as we've shown that the deletion of this protein is important to target TFAB in the nucleus. We can also try maybe these other therapeutic strategies in order to obtain the same phenotype.
Thanks.
Are there any other questions from the jury or from any of the attendees? If not, I would like to declare that the presentation part of our symposium is now closed. And upon closing this presentation, and I really would like to thank the three presenters, really outstanding, clear presentations, and also they really made hard the job of the reviewers of the different applications to select the finalists, and many outstanding application. And I really convinced that the choice of focusing on neurodegenerative disease was really a good one. And I am pleased to see how many young investigators are keeping this topic in their sight and in their focus, because there's a real need to enhance research and treatments in this field. I think without any further ado, I can give the floor back to Cinzia for some remarks, I believe.
Thank you, Alex. Great work. Thanks to the presenters, the jury. It has been, again, an exciting series of presentations that we have heard. We are very grateful to the embassy for sponsoring this award with a topic that varies annually, to always focus on something cogent and important for a larger segment of the community. And so in this light, it's now my pleasure to offer the opportunity to Dr. Antonella Incicchiti, who is representing the embassy tonight, to give some remarks. Antonella, you have the floor.
Thank you very much. Do you hear me?
Yes.
Okay, yes. So thank you to all. ISNAF is strongly supported by our embassy and also by the Italian Ministry of Foreign Affairs and International Cooperation. And you are making a very good job with our diaspora of Italian scientists in USA. And we thank you a lot for this. But my best compliments are for the finalists, the young scientists, for the brilliant researchers, and also for their passion for science that is very transparent from their talks. So good luck for the prize and for your future. And I'm looking forward to see you in our embassy on November 8th for the award ceremony. So thank you to all.
Thank you, Antonella. And with this, I thank you all one more time, and the meeting has come to an end. Thank you all.
Thank you very much.
Thank you.
Thank you.
Thank you.
Goodbye.
So much.